It's great to be here. Really appreciate the invitation to talk to you today about our TGX-DDI transcriptomic biomarker, leading from its development through some of the validation work that we've been done and hopefully promoting the adoption of this for regulatory decision-making to build a next-generation genotoxicity testing design strategy. I have the privilege of representing our team, our TGX-DDI biomarker team, which hails from the Health and Environmental Sciences Institute, or HESI, and specifically within its Emerging Systems Toxicology for the Assessment of Risk, or the E-STAR committee, shown here. I'm showing here the list of individuals who've been involved in the development of TGX-DDI for many years. There have been a lot of people involved, and I can't show all of them, but these are the sort of diehard TGX-DDIers. First of all, from Georgetown University, Dr. Heng-Hong Lee, who's been a leader in this program, right from the development, the original discovery of the biomarker right through to today. And our team here, shout out to my partners in Ottawa, Andrew Williams and Julie Buick. I'll show data that they've either analyzed or produced. And our fearless leaders, our management team over at HESI, Cyril Pettit and Chrissy Crute and Connie Mitchell. And of course, thank you to the agencies who funded this work over the years. Health Canada's Genomics R&D Fund, the U.S. Food and Drug Administration, the Natural Sciences and Engineering Research Council of Canada, and Canada Research Chairs Program more recently. We're here to talk about carcinogenesis, and I don't think anybody would argue with me that we all agree that genotoxicity is a key driver of carcinogenesis. So when DNA damage is inflicted on the cells, if it's not repaired, it can lead to mutations or chromosome damage. And this can confer selective growth advantages to cells that can lead to tumor formation. And we've known this for decades. And that's why we actually have an excellent test battery of genetic toxicology tests via the OECD. And there's even in vitro OECD tests. So I would say the genetic toxicology community is they're really pioneers in NAMs. So we currently use the following in vitro OECD test guidelines: the Salmonella bacterial assay, or the AIMS assay, the in vitro mammalian mutagenicity assays like the HPRT and TK gene mutation assays, as well as for chromosome damage, the in vitro micronucleus test. And as I said, these assays have served us well for decades, but they certainly do have their limitations that we must think about. So first of all, they lack efficiency. They are standalone tests and aren't easily integrated with other tests. But really importantly, they lack specificity. And what I mean by that is that we tend to have a high rate of positives in vitro that aren't reproduced in vivo. And so we call these irrelevant positives. Some people call them misleading positives or false positives. But this is an important limitation of these in vitro assays. And we need to have an understanding of mechanisms of action in order to be able to understand relevance to humans and probability that these effects in vitro will be converted into effects in vivo. So we're interested in the development of transcriptomic biomarkers in my group for a variety of applications. And so I just wanted to start generally with some information on how the methods that we use to develop transcriptomic biomarkers in our lab. So we begin with developing a library of chemicals that we call reference or training compounds or prototypical compounds that we know trigger specific mechanisms of toxicity or specific hazards. So we have our reference training set here, and an example here would be we categorize well-established chemicals that are genotoxic, non-genotoxic, or we might have some irrelevant positives in here. Then we do some exposures to cells in culture, and we'd run transcriptomics experiments on them to create the transcriptome, and we do some data processing on that. And then we'd work with our bioinformatic partners to identify transcriptomic biomarkers from this. So they'll apply supervised learning or machine learning approaches or curate sets of genes that can help us to identify gene sets that accurately predict one of the categories of chemicals that we have in our training set. They'll do internal validation exercises on these, things like cross-validation, leave one out analyses, to determine the stability and predictivity on these training set data. But really importantly, after that, they'll take them to external validation. So that's testing on data from outside that original reference set to establish the performance of the transcriptomic biomarker, its accuracy, sensitivity, and specificity. Following that, we would work on case studies with different end users to apply the biomarker in their specific contexts of use. So we can work with different sectors, making different types of decisions, and understand their context of use for this potential biomarker and how it works in their hands. This also facilitates technology transfer so that we can do ring trials. At this stage, we need to lock down our standard operating protocols and our data interpretation framework. And hopefully, the goal, of course, of all of this method development is to have some sort of guidance developed in the end. So let me introduce then our TGX-DDI transcriptomic biomarker. This is RNAM. It's an in vitro transcriptomic biomarker that predicts the probability that a chemical is DDI, which stands for DNA damage inducing, or non-DDI, using toxicogenomics. So that's where the TGX comes from. We developed this transcriptomic biomarker originally using DNA microarrays in human TK6 cells. So this is a very standard cell line that's used in genetic toxicology and a number of the other assays, so widely used and widely accepted. The TK6 cells were exposed to 28 reference chemicals that were categorized as being well established to be DDI or have clean genetic toxicology profiles, so they're non-DDI. And from that, machine learning identified 64 genes that are predictive of DDI potential. And you can see that in the heat map over here, where genes are on the y-axis and chemicals are on the x-axis, binned into DDI or non-DDI reference sets. And you can see the very specific pattern that differ between DDI and non-DDI. So that's our transcriptomic biomarker. Why do we like it? Well, we love using transcriptomics, and having biomarkers removes the subjectivity associated with transcriptomic data analysis, things like trying to understand what a change in a pathway would do. This is a much more objective approach than that kind of an analysis. We also like that these are endogenous cellular responses to DNA damage in human cells, so they're highly biologically relevant. And this endogenous response is a common characteristic of many different cell types, so it's transferable to other cell lines. We're using generally global transcriptomic approaches, and so we're just using this subset of 64 genes, and therefore we can look at other biomarkers at the same time. So we have multiplex capacities when we use these kinds of approaches. And what we like the most, and I'll show you some data to support that, is that the biomarker has a high specificity in that it correctly identifies these irrelevant chromosome damage results. So it classifies chemicals that we know cause irrelevant chromosome damage results as non-DDI. So this is how it works. We expose our TK6 cells to our chemicals. We do concentration response designs. We recommend lots of concentrations. We recommend a concentration range setting, first of all, that would be selected based on cytotoxicity at 24 hours. And we follow the guidelines for the micronucleus in vitro test, where we go up to 10 millimolar of testing or solubility to concentrations that induce about 55% cytotoxicity, plus or minus 5%. So around 50% cytotoxicity. And that's because genotoxicity happens in kind of this narrow window, and you have to push it up to almost these cytotoxic concentrations in some cases in order to be able to get to see the DDI response. So we expose the cells either in the presence or absence of S9 microsomal fraction. This is because the TK6 cells aren't metabolically competent. They're exposed for 4 hours. With S9, we have a bit of a recovery period. And then the cells for transcriptomic analysis are sampled at this earlier time point. We did this work initially on DNA microarrays, but we have since transferred the technology, and you can see all the different types of transcriptomic profiling platforms that we've tried and worked successfully for TGX-DDI. We get the data, we do some bioinformatic processing, and then we apply our classification strategy. So there's 3 steps in the classification strategy. The first is called a nearest shrunken centroid probability analysis. So this is a probability that your new chemicals pattern actually matches either the DDI signature or the non-DDI signature with a probability of greater than 0.9. So if it's less than 0.9, it falls somewhere in the uncertain range. We do a principal component analysis alongside the reference training set. And in this analysis, if you have a negative principal component 1, you're classifying the chemical as DDI, and a positive, it's non-DDI. And similarly, there's a two-dimensional clustering approach with the reference chemicals, and you see which branch it falls on with the DDI or the non-DDI. And that allows us to differentiate between DDI and non-DDI agents. And what we'd say is that, to be conservative, if you have a positive in any one of these three classification approaches, you would call that DDI. It has to be clean negative across all of them to be non-DDI. To make this easier for those of us who don't have bioinformaticians in our lab, we worked with the NICEATM, specifically with Jennifer Fostel, and she created a really nice, easy to use TGX DDI biomarker classification tool where you just upload your gene counts and the tool does all the work for you to classify the chemicals. All right, now we have a couple of different contexts of use that we've considered, and I'll show you an example more of the chemical safety assessment and walk you through the ring trial for our context of use number one. So first is drug screening. So the Health and Environmental Sciences Institute's biomarker working group initially was very interested in this area specifically. And so this is to address the low specificity of the chromosome damage assays. And so in drug screening, if you have a drug candidate and you have a negative Ames assay, but a positive chromosome damage assay, and you paired that with an in vivo micronucleus test that's negative, then you're kind of left in a situation where you're not sure what to do with the chemical next. So we need to know if this in vitro chromosome damage assay is relevant or not. So at this point, you would apply the TGX DDI biomarker to differentiate between potentially misleading results and a relevant positive. So that's context of use one, where we applied the TGX DDI in a weight of evidence analysis to inform irrelevant in vitro chromosome damage results, and this was brought to the FDA. The second is more my area, so chemical safety assessment, and this is really about using TGX DDI in integrated testing strategies to inform potential hazards, mode of action, and even for potency comparisons. And I'll show you a result of this, but we really think integrating TGI with the other NEMs that are available within the GeneTox community is a really powerful approach to help us move towards reducing animal tests. All right, so I'm going to answer some of the questions that were provided in the template. First of all, speaking directly to the biological relevance, we've already discussed how genotoxicity is a key driver of carcinogenesis. But what I'd like to mention further about mechanistic understanding is that the TGX DDI biomarker is enriched in genes that are under the regulation of P53. About 75% of the genes in our biomarker are directly or indirectly regulated by P53, which is a really key nuclear receptor involved in the DNA damage response pathway. It also measures intact endogenous genomic responses to DNA damage inducing agents in human cells. So highly biologically relevant biomarker. We have tested it with a lot of different reference compounds now, and we observe high concordance with our expected results using these reference compounds and high concordance with other genotoxicity tests. So let me show you an example of some of the work here. In this validation study, we had chemicals in 5 chemical classes. I'm only showing 3 of them here for simplicity. Class 1 are direct acting DDI agents. Class 4 are non-DDI agents, and class 5 are these problematic, irrelevant in vitro positive chemical agents. So you can see here, these yellow squares indicate a positive result in the chromosome damage assay and a positive in the Ames assay, whereas blue is negative. And here, these little blocks represent for each of these chemicals in class 1 through 5, whether they classified as DDI, yellow, with our 3 analyses and the overall classification. So if we just look at the overall here, we can see we've correctly identified all of the class 1 agents as positive. We've got 9 out of the 10 correct results for the non-DDI agents. And again, we have a nearly perfect class 5 classification. So the results of this study show that TGX DDI actually quite effectively predicts these class 5 agents with high sensitivity and specificity. But we want to move towards more quantitative approaches as well in the genetic toxicology area. And so one of the things that we like to do is to use this biomarker for potency comparisons and for comparing against other biomarkers. And so in this experiment, which was actually, it's done by Julie Buick, you can see the reference down there. These were HEPA-RG cells that we exposed to, I think it was 10 prototype agents. And we ran the TempoSeq transcriptomic profiling platform on these cells. So in the experiment, we derived a gene set benchmark concentration responses for all the positive responding chemicals, all the DDI agents, for the TGX-DDI biomarker. And how we did that was every gene in the biomarker was fit to a dose response model, different dose response modeling curves to identify the benchmark dose or the concentration at which we see a defined increase relative to the control samples. And then we take all of those gene BMCs and collapse them into a median with a confidence interval associated with them. And then we compare that to damage measured using the COMET chip, which measures single strand breaks and abasic sites and alkaline labile sites. And when we do that for the compounds that were positive, we see this beautiful correlation. This is the COMET benchmark concentration on the y-axis and the TGX-DDI biomarker concentration on the x-axis. So really nice concordance between the results and identical potency ranking between them. So high concordance both with the chemicals that we expect to see DDI and non-DDI, and also with other genotoxicity tests. So we've characterized over 100 chemicals now in different cell lines using different transcriptomic platforms and using some variations of study designs. And we've learned a lot over the years by doing this. So first of all, in terms of sources of variability, an important one for the TK6 cell line is the S9 lots, which can have variability between batches. Different lots of cells. So every time, you can't rely on an old test concentration that you've set. A new batch of cells may give slightly different responses. So you always must do a concentration range setting. You can use the TGX-DDI biomarker in different cell types, but the cells that you use must have an intact p53 response. We've got sources of variability associated with common areas of variability in genomics. So for example, RNA quality must be high. And I've already mentioned the need to test up to a certain level of cytotoxicity. So if you adhere to our protocols, we get very high performance standards. Again, top concentration should approach 50%. If you've switched to a new cell line, you must first do a bridging study that identifies your optimal sampling time and apply some quality controls to make sure that you've got high quality, both RNA as well as the quality of your sequencing or whatever you've done is also high. If you do that, then our methodology is very robust, and our bridging studies have demonstrated accurate and sensitive results for TGX-DDI in a number of HEPA-RG cell studies and across multiple transcriptomic profiling technologies. All right, now a few slides. In the next few slides, I'd like to tell you about our FDA biomarker qualification study. So we brought this biomarker to the FDA many years ago. And so the study design that I'm going to show you now reflects years of conversation with them and lots of fantastic input. So the objectives of the qualification were to assess the cross-lab reproducibility of TGX-DDI classification calls using one platform, NanoString, at 4 sites and using 13 chemicals alongside 4 controls. So each site received the test compound as a stock solution. They received 3 pre-designated concentrations that they should test and the solvent control. They used TK6 cells. Everything was done in triplicate, each concentration, and everything was done following the locked down standard operating protocol that we provided to them, including the analysis and the data processing. And they used the NIHS's classification tool to determine if the compounds that they used were DDI or non-DDI. So this is really exciting because I have brand new data to show you that's really hot off the press. These are very preliminary results. We haven't shown the FDA yet. Here's our compound codes here. And you can see what the expected DDI and non-DDI calls would be. And here's our positive controls down here. And what we see is that our biomarker is performing exactly as expected in the labs. What we did learn was the concentration range setting. As I mentioned, this is one of the things that we learned from this. Some of the labs did not achieve a high enough level of cytotoxicity in the assay, and thus we could not call the tests valid. So that's why not all of the compounds have a 4 here as the number of tests conducted. Nevertheless, of those tests that were valid, we can see we were 100% sensitive. We correctly identified all the DDI agents. We had a few false positives, in our non-DDI agents, and we classified our control chemicals exactly as expected. So overall, that led to an accuracy of 93%, a sensitivity of 100%, and a specificity of 86%. So we're quite excited about that. Right, so that is the technical validation, the biological relevance. Now let me just finish with an example of how we are using the TGX-DDI biomarker. So this work was done in Health Canada with my long-term collaborator, good friend, Dr. Paul White, who's the lead of the Genotox 21 program there, and with a fantastic student that we co-supervised, Anne-Marie Fortin, who was an MSc student with us and is now a biologist at Health Canada. And this was to apply a case study for data-poor new substances at Health Canada. So you can see the paper here that was published last year in Frontiers of Toxicology, and all the authors involved here. So the objectives of the study were to explore the integration of TGX-DDI within Health Canada's Genotox 21 platform to predict the genotoxicity of data-poor chemicals on Canada's in-commerce list. So these are chemicals that were given to us by our partners in the New Substances Assessment and Control Bureau. So you can see NSACB compounds 1 through 10 here. And they had prioritized these chemicals as requiring genotoxicity tests because of flags on QSAR. So these compounds are present in cosmetics and hair dyes, cleaning formulation, and some of them are pharmaceutical intermediates, and they're listed here for you. And Anne-Marie also included 4 positive control chemicals. So the Genotox 21 platform is an integrated platform with a number of different NAMs in it. And Anne-Marie's project, she decided to use 3 of these NAMs. First of all, the microflow, which is an in vitro micronucleus test assay. And this one we really like. It's fast and works really well, but it has very high sensitivity and lower specificity. So she ran all the chemicals first through the microflow assay. You can see that the positive and negative chemicals responded as expected, and 9 out of 10 of these compounds classified as positive in the microflow assay. Then she ran the TGX-DDI and the multi-flow assay. So multi-flow is a flow cytometry-based assay available from Litron Laboratories, and it has a bunch of biomarkers for things like P53 activation, gamma H2AX, and mitotic indices. So this pairs very nicely with TGX-DDI. And what we see when we do that is, first of all, we're happy to see that compound 10 is negative here across the board. So we've got 1 NSACB compound that's negative for every assay tested. We see a number of reds all the way across, so we can conclude that these are likely genotoxic compounds. And the multi-flow assay gives us additional mechanistic information to indicate that these are likely clastogens. That's what the +C means. But what we also see is some negatives, some discordant results with the microflow assay. And we argue that these are the ones that the TGX-DDI and the multi-flow do a good job of capturing these irrelevant positive results. And so this case study allowed us to come up with a data interpretation framework for combining these assays in Genotox 21, where we have data-poor chemicals and we run the microflow assay, and we can separate the chemicals into micronucleus-inducing or non-micronucleus-inducing. Then we apply the TGX-DDI biomarker to understand if the micronuclei are caused by DNA damage or not. So if we've got DNA damage, positive responses by TGX-DDI, then we then apply the multi-flow assay to differentiate, if we need to see if it's a clastogen or a pan-genotoxic chemical. If we have a positive micronucleus test but a negative non-DDI, there's 2 potential outcomes that the multi-flow can help in tie-breaking here. These compounds could either be antigens or they could be these irrelevant positives. And so this is how these assays integrate together to provide our really effective NAM-based strategy for genotoxicity assessment, in particular for these data-poor compounds, but for other contexts of use as well. All right, so to wrap up then, I hope I've convinced you that our TGX-DDI biomarker has been extensively validated, and it really does accurately predict the potential of chemicals to induce DNA damage across several different cell lines and transcriptomic platforms. And happy to announce that we are very close to the final stages of FDA biomarker qualification. I've only shown you one case study, but there are others. And if you want to get in touch with me, I can point you to those so you can read about those as well. They've been very useful in informing regulatory applications and building confidence in the use of TGX DDI. And they demonstrate how integrated testing strategies incorporating TGX DDI are effective to inform potential genotoxic hazards, mechanisms, and for potency comparisons. And there's a lot of great transcriptomic biomarkers out there that really are providing these sort of efficient and objective ways to analyze transcriptomic data. So we hope that our biomarker serves as an example as to why they're so valuable. And we think that collaborations would be really useful to help us identify the best path forward to promote regulatory uptake of these transcriptomic biomarkers. And I highlight that and emphasize that particularly because the story that I'm telling to you now has been over 10 years in the making. So thank you very much for your attention. There's a QR code here to learn more about the work that's important that's going on within the eSTAR committee on all of the NAMs and other things that are happening, including TGX DDI within eSTAR. And if you'd like to get involved, there's still lots of opportunities. So please check out that information, or you can just try TGX DDI on either existing or new transcriptomic data from human cells and culture. And we're certainly happy to help with that. And my contact information is right here. So thank you very much for your time and attention.